Hello and a very good afternoon to Dr. Go and Dr. Peter and also fellow students. We are from Group 6. Are going to present about the glucose oxidase. Our members consist of me, myself, Mardila, Tivashini, Kartini, Sitish, Saraza, Suganya, and Jennifer. And for the first task is about the classification of properties of glucose oxidase from Aspergillus niger. The classification is GOX is an oxidoreductase. The EC number is 1134. The synonyms is beta-D-glucose oxygen 1 oxidoreductase and also glucose oxyhydrase. The catalytic reaction is beta. It catalyzes the reaction of beta-D-glucose and oxygen gas to D-glucanol 1,5-lactone and also hydrogen peroxide, and also GOX are usually found in the mycelia of Aspergillus niger. The physical properties of GOX is shown in the slides. Molecule with a molecular mass of 65 kDa, pi of 4.2, and the GOX is found as a homodimer protein. And the catalyzation of GOX requires FAD as the cofactors. The enzymatic properties of GOX is that the specificity of GOX is for beta-D-glucose with Km of 33 until 110 millimolar. The similarities and differences of GOX are found in two different organisms such as Penicillium species and also Aspergillus niger. The similarity is that both GOX are the GMC oxidoreductase family, and the differences is that Penicillium species, the GOX of Penicillium species has less stable but has higher affinity for beta-D-glucose and higher specificity constant than Aspergillus niger. It also has the higher activity and affinity in Penicillium than Aspergillus niger. For the next task is going to be presented by my co-presenter.
I will present about industrial application of GOX. As we know, GOX is the most widely used enzyme. The first application is used as glucose biosensor for diabetes monitoring, and also as a biofuel cells, which is convert biochemical energy into electrical energy, and also used as a commercial source of gluconic acid. And GOX also will react with lactoperoxidase to be used as an antimicrobial agent in oral care product, and applications in the textile industry as a method for producing H2O2 for bleaching. GOX also used to remove residual glucose and oxygen in foods and beverage in order to prolong their shelf life, and also to produce polymers like propylene bromohydrin. And YSI analyzer designed for use in clinical chemistry and for formulation in an ink suitable for printing and the fast and efficient electron transfer. Next, my first.
Okay, I will explain about the protocol to measure the enzymatic activity of GOX for different substrates. The method we use is continuous spectrophotometric weight determination. The condition is 35 degrees Celsius and 5.1 pH. The reagent we use is sodium acetate buffer, O-diacidine solution, glucose, fructose, galactose substrate solutions, peroxidase enzyme, and glucose oxidase enzyme solution. Okay, this is the method we have to do. Okay, the first pipette the following reagent in suitable cuvettes, then add the glucose oxidase and buffer, then we measure the absorbance value. The same step goes to different substrates, fructose and galactose. We use this method because it is suitable to assay for glucose oxidase activity and kinetic parameter by using either cell-free extract or enzyme, purified enzyme.
Good afternoon. Let's take a look on how we can extract GOX from Aspergillus niger. Our group used bit beating method because this method enabling microorganism is slow from the implant but not be killed. Second one, it's reliable and fast method not restricted by the species or stage of development. This is the protocol on how we extract the GOX from the Aspergillus niger. First, we add buffer to the sediment and then we incubate and vortex. After that, the sample we put in with tubes and then homogenize for 3 minutes, and then we centrifuge. After centrifuge, we extract the supernatant and store on ice. After that, we wash the pellet and with buffer and repeat with centrifuge, and then we combine the supernatant and add equal volume of phenylchloroform. Mix, centrifuge and repeat at last one more time until the phase is clean. And then we continue centrifuge and discard the supernatant and wash with 80% ethanol and resuspend in distilled water and store the DNA at negative 23 degrees Celsius. Thank you.
Okay, next is how to produce purified GOX. The first step is the enzyme fraction was centrifuged, then precipitated with ammonium sulfate. Then the protein precipitated will centrifuge again, then purified with gel filtration. Then the enzyme fraction was full and further purified with ion exchanger column. Next, glucose oxidase elute a single peak from ion exchange column. Okay, the methods that we use is protein precipitation. Ammonium sulfate is added to precipitate the protein by altering the solubility. The reasons are to stabilize the protein precipitate and remove undesired contaminants. Next is filtration. Centrifugation separates samples by spinning them at high speed. So this protein will be isolated into supernatant and pellet. The reason is to separate molecules based on size and density. The last method is chromatography. We use ion exchange chromatography and gel filtration chromatography. The reason is to sort protein based on net charge and to sort protein based on size.
Okay, next I would like to explain how to demonstrate the purified protein is GOX. Firstly, we can use SDS page. The main purpose of SDS page is to determine the molecular weight of GOX. The molecular weight of GOX can determine by denaturing the SDS page in polyacrylamide gel that containing 2-mercaptoethanol, and then the protein band revealed by staining with co-emissive brilliant blue and silver nitrate. Next, compare with the molecular weight. The molecular weight was calculated by calibration against phage ruler. Therefore, the molecular weight of GOX from Aspergillus niger is 131.28 kilodalton. Second step, we can use Western blot. Western blot is used to detect the specific protein, which is GOX in purified protein. Next, we can conduct glucose oxidase reaction mechanism. Firstly, GOX will catalyze the oxidation of beta-D-glucose to D-gluconodelta-lactone and H2O2. So the final product will be water and oxygen. Next, how to demonstrate that there is no significant contaminant in sample. First, we can use electrophoresis if the purified GOX contaminate by DNA. If there is a band, it may contaminate by DNA. If there is no band in the gel, it means there is no DNA. Next, we can do chromatography if there is a virus in a GOX protein.
Next is about want to investigate the 3D structure of GOX and identify the active site of this enzyme. First, this is the structure of GOX that we found from Niosara. And method that used is bioinformatic tools, which is UniProt. UniProt provides the central database of protein sequence and annotation. Then use Clustal W2, a multiple sequence alignment tools for the alignment of DNA or protein sequence. This will provide identification of conserved sequence region. Reason why you use these bioinformatic tools because it is high quality of database, accurate and annotated protein sequence, freely accessible, easy to interpret report, and save time.
Okay, structure of denature enzyme can determine by X-ray crystallography. Okay, this is the overview. Okay, we choose this method because we could get the whole 3D structure accurately compared to other methods. Okay, that's all from me. Thank you.